Hello, everyone, and welcome to week 3. I hope you're having a great start of the semester. I want to start by saying that next week is a 4-day weekend. So Monday, September 7th and Tuesday, September 8th are a holiday and a reading day. So I hope you have a really nice long weekend, maybe catch up on some of your classes, including Colloquium. So we're on the week 3 module. Make sure that you're working through everything that's assigned there. And we're going to be continuing with our discussion on phytoremediation and starting on bioremediation. So these are my 2 areas of research. I find them fascinating. We're using plants and microbes to solve the world's problems, clean up chemicals, make the world healthier and safer and more sustainable. So last week I was discussing how we can use plants in phytoremediation. But one of the things I didn't really touch on was some of the difficulties in using plants to clean up chemicals. So these are some photos from a job site that I actually worked at. And you can see on the left-hand side, this tree is having a very hard time. It's all brown and it's not surviving very well. So we found this was actually from boron toxicity. There was too much boron in the water that was being used to irrigate the trees, and it was causing the tree to die. So the chemical itself was not harming the tree, but just some chemical in the water was making the tree die. And when the tree dies, it's not able to remediate the chemical, and you still have the same, hazardous cancer-causing chemical at the site that you did in the first place. On the right-hand side, you can see a picture of a really cute little ladybug. We actually had to bring in ladybugs because we had an aphid infestation on our trees, and the aphids were infecting our trees and really causing them a lot of stress. So we brought in these cute little ladybugs instead of trying to use pesticides, and ladybugs eat a ton of aphids. So we were able to successfully kind of manage that situation, but it took a few months and slowed down the tree growth. And then lastly, we had borers, which are these maggoty looking bugs that burrow into the xylem of a tree and restrict the sharing of nutrients and water. So it essentially steals the nutrients and water from the tree to grow itself. It grows big enough that it can eventually change into a moth-like creature and fly to another tree. But in doing so, it grows inside the tree and causes the tree to die. So we would have to go through and find trees that were infected with borers and try to kill them. We tried using nematodes, which are kind of like little worms, to eat the borers, but the borers were a little too good, and eventually we had to use a pesticide, but we still have had issues with them coming back year after year, and they can just devastate tree crops. So those are some issues that I've noticed in my personal work with phytoremediation. There are other issues like bunnies and squirrels eating the bark, flooding, drought. These are biological living trees. So any of the issues that we have every day using trees for things such as yard plants can be bigger issues when you're trying to use them in engineering applications or sustainability practices. So that's kind of one last thing I wanted to talk about with phytoremediation, summing up last week's lecture where I introduced different ways of using phytoremediation for cleaning up hazardous waste and different chemicals. But now we're going to switch to bioremediation, which is another type of sustainable remediation. And instead of using phytoplants, we're using biomicrobes. So here in bioremediation, we're using different bacteria, fungi, sometimes viruses, not really. It's mostly bacteria and fungi to remediate chemicals, just like in phytoremediation. Part of phytoremediation actually is bioremediation, since the plants have microbes that live inside the plants and on their roots. So in bioremediation, a lot of times we as researchers or governmental agencies will identify different microbes that can degrade a chemical. So for instance, if you have a starting chemical like the one I mentioned from former dry cleaners, PCE, and you have a microbe that can degrade that all the way down into a sugar by itself, well, then why don't we just dump all these microbes at the site with the PCE? And that's kind of the thinking behind bioremediation. First, checking if the microbes can survive, checking if they're dangerous to people, checking if we can control them, and then if they make any byproducts on the way to creating a sugar. So one of the big issues is that a lot of sites have a lot of contaminants. So a lot of times we're not looking at just one chemical. You know, if you're looking at a landfill or a site where they're processing a lot of things or treating a lot of things, you don't have one cancer causing chemical. You don't have only benzene, or you don't have only PCE. Most of the time you have a mixture of a lot of them. And so finding microbial groups is really important instead of finding just one microbe, or potentially focusing on one chemical at a time. But sometimes these chemicals can be toxic to other microbes, or they can create unintended consequences. One of the cool things, hopefully you guys can see this, the EPA runs a website called, it has a website, EPA does, and then it lists all the Superfund sites throughout the United States. So Superfunds are sites that have been identified, there's about 4,000 of them, as being contaminated. So they are posing a public health threat by their levels of chemical contamination. And you can look at the state that you're from and see if you can see any Superfund sites or national priority sites, these really heavily contaminated sites that are being forced to be remediated. The government is saying, you have to clean this site up. Whoever's owned it or owned it before, you have to clean it up. So there's a lot in Florida. There's a lot over the United States. I encourage you to look and see if there's any in your hometown or anywhere near you. I went ahead and looked up some that are near us here in Fort Myers. So there is a site that's not currently on the list, but it could be, and it might almost be. It's on Route 50 next to Arlington National off Lee Avenue, and it's a site that's undergoing cleanup right now. So I don't really know much about this site. They don't have a ton of information, but I pulled it from this website, Home Facts, that allows you to map this area and see where there's different gasification plants, former Superfund sites, active Superfund sites, almost Superfund sites, and different areas of concern. So a lot of people use this if you're buying a house, but it's also interesting to just look at kind of what's nearby chemically. When I was in consulting, I worked on a lot of these sites, Superfund sites and national priority list sites. Some of them are in the desert, some of them are in old former buildings, some of them have been vandalized or abandoned for a while. So you kind of get a wide variety of sites that you're doing bioremediation at. It's not just in the lab, and it's not just at these beautiful, contained little areas. Sometimes they're huge, sometimes they're in buildings. And the way that we choose to remediate chemicals at a site, we don't always choose bioremediation, but the way that we choose to remediate chemicals at sites is based on a lot of things. First, what hazardous waste is there? What are the chemicals that are there? Is it a chlorinated solvent, or is it a, TNT? And we can kind of choose what options we have for remediating those chemicals once we identify what they are and what concentration they are. In addition, where is the site? Is it near a city? Is it in the middle of a desert? Does it have a lot of rain? Does it have a lot of vandalism? What are the site conditions that are there? I worked on one site where there's no cell phone service. We were about 3 hours from any significant airport, and it was really hard to get people and trucks to the site. So maybe having a big, intense machine remediation wouldn't be a good idea at this site. Also, time and money. Money and time are always considerations in pretty much every project you'll ever work on. So they're also a consideration on these projects. But overall, when looking at chemical remediation and cleaning up metals and different chemicals or TNT, a lot of times bioremediation is one of the cheapest and one of the lowest impact. So it doesn't require huge buildings or large trucks a lot of times. It can be pretty low impact and low cost. Compared to other methods, like I'm not going to discuss these and you don't really need to know them, but compared to other methods, it is significantly cheaper. Bioremediation is a general term when we're using microbes to remediate chemicals, but there's actually a lot of different types of bioremediation. Natural attenuation is what's just happening naturally. Inherently in the ground, microbes are already degrading chemicals. So that's just what they're doing naturally. And sometimes that is a course of action. If you leave a site alone for 20 years, it'll kind of clean itself up sometimes. There's also biopiling and land treatment, which is kind of like composting. Bioscrubbing, where we a lot of times use these filters with microbes on these filters. Bio walls and bio barriers, where if we have flowing water, like in a river or groundwater, we'll put up a wall that has microbes in it. And as the water flows through it, it gets treated. Bioreactors, where we have kind of a big metal reactor and we put in the contaminated material and we add microbes and we let them react in this contained system. And then there's bioventing and biosparging, which is where we're injecting air or oxygen into the ground to allow microbes to have an electronic acceptor and enhance their activity. So there's generally two types of bioremediation, the top being biostimulation, where we're just adding oxygen or nutrients that are limiting the microbial growth. So a lot of times nitrogen, phosphorus, or oxygen are in limited supply, and they control how much activity the microbes can have. So if we add a lot more of these and stimulate the microbes to grow, we should get more chemical degradation. There's also bioaugmentation, where instead of adding just nutrients, you're also adding microbes. So say if you discover a microbe that degrades plastic and you have a big landfill with a ton of a ton of plastic, you can take this microbe and put it at the site for it to degrade plastic. Or if you find that there's already microbes at the site that degrade plastic, maybe you just feed them a little more food and see if they act faster. Here is a nice kind of overview of how we have natural bioattenuation, where we just have the microbes that are at the site acting at their own rate over time. You have biostimulation, where you're adding fertilizer, water, carbon, nitrogen, or phosphorus. This is making them speed up and act faster rate of removal of these chemicals, like a pesticide. This can be any chemical here. The chemical and microbe will change based on sites, obviously. And then bioaugmentation, where you're adding not only bacterial or microbial strains, but you're also adding some of the nutrients you would in biostimulation. So you're adding these exogenous microbes from another area or another site that aren't inherently at that site. What it looks like when you're doing this, a lot of times there are times where we might spray the microbes down, but really we'll either mix them in big reactors, or we'll inject them into the ground in groundwater as shown here. So KB1 is a microbial consortia that can degrade PCE, that dry cleaning chemical that causes cancer. That's a lot in groundwater. So here we are just injecting it down into the ground. We're just adding microbes right into the ground, right into the groundwater. And here's an example of my friend and I actually doing this at a site. So we're doing some water sampling at an area where we've already injected in a bunch of chemicals. So you have these wells, you inject in a bunch of nutrients or microbes, whichever you're doing, biostimulation or bioaugmentation, and then you monitor over time how the chemicals are degrading. So if you live near a Superfund site or a former Superfund site or an area of interest, and you hear about phytoremediation or bioremediation being done at those sites, I hope you have a little more interest and knowledge about what might be happening. As you can tell, I love this area. I dedicated my career towards looking at ways we can clean up the environment using microbes and plants, and I just think it's fascinating. So please email me with any questions or comments or anything you'd like to discuss, and I hope you have a great rest of your week. I'm gonna see if I can stop screen sharing. Let's see. All right. With that, I will see you all next week. Have a great long weekend and great rest of your week.